Good afternoon, everyone. So thank you very much to the organising committee, first of all, for organising such a fantastic meeting and also for the opportunity to present our project. So newborn screening model using integrated multi-omics in South Australia, abbreviated to NewbornsInSA. And I'm one of the clinical leads on this project, and I'm presenting this on behalf of our wider study team. So we are based in Adelaide, which is the capital city of South Australia. This is a lovely city. South Australia is home to just under 2 million people. And SA Pathology is a public organisation, our public pathology service that currently provides standard of care newborn screening to babies born in South Australia, also part of the Northern Territory and Tasmania at the present time. So in accordance with SA Pathology, we work very closely with our clinical centre, the Women's and Children's Health Network, and so SA Pathology, the Women's and Children's Hospital, and then also the University of Adelaide are the three major partners involved in this program. And our team is led by Principal Investigator Corinne Kassan, and we have a large number of chief investigators on the project, spanning clinical medicine, laboratory scientists, our health implementation team, and our project manager, and many of us work at either one or two or even three of these different organisations. So we're really uniquely placed to work together very collaboratively in a pre-existing system which provides standard of care newborn screening, and it's a great place to essentially then expand this into our research project. So essentially this is a pilot study which is looking to expand current screening capabilities to identify over 500 different conditions. And essentially we're evaluating a novel multi-omics approach which will integrate a combination of metabolomics, whole genome sequencing and also clinical information through which we are hoping to identify at-risk newborns. So Lillian had mentioned before our GenScan consortium, so our local Australian group, so I won't go into too much detail about this again. And I guess just to mention that genomic newborn screening really is seen as a priority area for research by the Australian government, having funded our 5 different projects in the order of 15 million Australian dollars to undertake further work into this field. And the GenScan consortium is really a great group, and it's bringing us together really to share experiences, eventually to share our results, and ultimately to have a unified voice which will then hopefully inform national policy moving into the future. So essentially our study design is a prospective return of result study where we're returning results in real time. We will be aiming for results to be returned within 4 weeks, and we will be using current Hill-Prick blood samples on dry blood spots, so no additional samples will need to be collected. We will be recruiting families during pregnancy and the first 2 weeks after birth, and essentially we're hoping to recruit 40,000 unselected newborns for our metabolomic screen, and 1,000 of these will go on to have whole genome sequencing. So just a study overview. So the first part of our study is essentially a retrospective validation study, and this is where we're using metabolic screening, so quadrupole time of flight mass spectrometry, essentially to look for any outlier profiles in those newborns who have a range of different conditions, and to compare these to the healthy population in the background. And these will be validated against known cases, and this will be untargeted in the first setting, which will then inform a more targeted metabolomic approach in our prospective arm. A proportion of patients with known genetic conditions will also be validated through our whole genome sequencing pathway, so that we can essentially optimise our DNA extraction and all of the downstream reporting, and these will actually be reported blinded by our genomic analysts in order to assess this pipeline. So the second phase of our study is our prospective arm, and I'll just mention at this point that we will have very heavy engagement of various stakeholders in terms of the study design, various aspects of the rollout, and of course in terms of assessing acceptability and various other processes as well. So as mentioned, we'll be recruiting patients in pregnancy, 40,000 unselected newborns who will initially have a first-tier metabolomic screen. A proportion will then go on to have whole genome sequencing, and this a proportion will then have clinically actionable results, which will be reported to managing clinicians, which will enable further investigation and appropriate treatment. We'll also be recruiting some 300-odd patients who are babies who are identified to be clinically at risk based on prenatal factors, and also those babies who are essentially unwell in the newborn period who would then benefit from this additional screening in addition to standard newborn screening as is current standard of care. And our final part of this project is our health implementation and health economic analysis, and essentially this will be aiming to look at both clinical effectiveness, feasibility, and also cost effectiveness of this new approach as compared to current standard of care. And again, we hope that this information will all provide helpful advice to policy making in the future. So in terms of where are we now, so we're currently undertaking our retrospective study. So we have ethics and governance approvals in place. The metabolomic validation is currently underway, and so we have completed metabolite stability experiments and have found accurate and reproducible results from historic Guthrie cards that are up to 4 years old. We're also in the process of developing the statistical methods for identification of those key features that are outlined in the metabolomic profiling. We have identified and accessed various cards from those children who have known diagnoses, and also 4,500 background sort of control cards that have been selected. And we're also establishing our methods for various aspects of processing of the cards, and this is all underway. And I would just like to take the opportunity to acknowledge our newborn screening unit team, Emily, Carol, Thomas, Alex and Kwa, who have worked very hard on getting this all up and running. In terms of our prospective study, so we're now in the final stages of our ethics application, and essentially this is for our prospective cohort, and we're also working on our stakeholder engagement as part of this as well. Importantly, we're looking at ways of recruitment, and it's been very helpful to hear some of the discussions around that today, over the last couple of days. But essentially we have developed a website, various information pamphlets, having a social media strategy, and are looking at setting up an enrolment database and various online questionnaires, and patient information and consent sheets. And we're also in the process of determining the metrics that will be used for the economic evaluation as part of our study as well. So in terms of the future, so we hope that by this time next year we will have completed our retrospective study, and essentially having validated our metabolomic screen as a first tier screen. We will have identified those key outlier features for the prospective targeted metabolomic assay as a screen, and also validated all of our DNA screening pipelines, which will be whole genome sequencing based. And in terms of the prospective study, we are also, obviously we'll have ethics and governance approved and completed all our initial stakeholder activities, finalised our communication strategies, and essentially we'll be hopefully be enrolling patients and screening them prospectively. So I would just like to take the opportunity to thank all of our partners and all of those who are involved in this research. These are our contact details if anyone has any questions or comments down the track. Thank you very much.